Imagine this, your heart beats nearly 1 lakh times in a day and your brain never sleeps. Your kidneys filter around 180 liters of blood daily. But have you ever wondered where does all this energy come from? Is it glucose, fat, proteins? No, they are only fuels. The real energy factory is hidden inside our cell, a tiny structure that is the mitochondria. And inside it runs one of the most beautiful processes in our human body, that is electron transport chain. Understand this one topic today and you will understand how almost 90% of ATP in our body is produced, and trust me, this is one of the most frequently asked in university exam topics. So let's begin. Before we start, let me ask you one question. If glucose gives you energy, why can't glucose directly produce ATP? Think about it. Instead, our body first extracts electrons from the glucose, fatty acids, and amino acids. Those electrons are carried by the special molecules that NADH and FADH2, and finally, all these electrons reach to the one destination, the electron transport chain. This is where maximum ATP of the body is generated. And so if you understand today's lecture, you will understand cellular respiration, ATP synthesis, cyanide poisoning, mitochondrial diseases, and oxidative phosphorylation. Everything becomes easier. Let's first understand the whole story before learning individual complexes. Here we have carbohydrates, fatty acids, and amino acids. These nutrients undergo different metabolic pathways and like glycolysis, beta oxidation, and TCA cycle. Eventually, they all produce two important molecules, that NADH plus and FADH2. Now here's the important question, are the NADH and FADH2 ATP? No. Think of them as a fully charged power banks. They are carry high energy electrons, and now these power banks reach to the electron transport, and here electrons are gradually transferred from one carrier to the another. As the electron move, energy is released and energy is used to produce here ATP. Finally, electrons reach oxygen. Oxygen accepts them and combines with the hydrogen and forms the water. So remember this complete flow: food, NADH, FADH2, then it goes to ETC, then finally ATP and water formed. This single flow summarizes the entire chapter. Many students write the glucose directly from forms the ATP, but that's incomplete. Actually, the glucose first generates NADH and FADH2. Most ATP is produced only after these enter in the electron transport chain. Why oxygen is important? Now let me ask you something. What happens when the oxygen suddenly disappears? Can electron transport chain continue? No, absolutely not. Oxygen is a final electron acceptor. Without oxygen, electrons have no here to go and whole chain stops. NADH cannot become NAD plus and FADH2 cannot become FAD plus. Whenever you hear ETCs, immediately remember oxygen is mandatory. Here, now we know the ATP is produced, but where exactly is the electron transport is located? Inside the mitochondria. Let's understand its structure. This is the mitochondria. It is the outer membrane. It is act as only protective covering. Now inside this, between the two membranes, here is the intermembrane space, and don't ignore this space. Later you will see it becomes filled with protons. That protons accumulation is the secret behind the ATP production. Next comes to the most important layer, that the inner mitochondrial layer. This membrane is highly folded and folds are called as cristae. Now here's the question, what is folded? Why it is folded? Exactly like a folding like a long paper to fit in the small box, folding increases the surface area. Greater surface area means more respiratory complexes and more ATP synthase and more ATP production. This is favorite Viva question. Now look inside. The central portion is called as the matrix. Matrix contains enzymes responsible for TCA, beta oxidation, oxidation of amino acids. So remember, most metabolic reactions occur inside the matrix, whereas ATP generation occurs mainly on the inner mitochondrial membrane. Now we reach the actual electron transport chain. The inner mitochondrial membrane contains the 5 respiratory complexes. Let's understand it each one by one. First one is the NADH dehydrogenase. This is the entry point for the electrons coming from the NADH. These electrons begin their long journey. Complex 1 is also pumps protons from the matrix into intermembrane space. Remember, this complex first performs the two jobs: transfers the electrons and pumps the protons. Second one is complex second succinate dehydrogenase. Notice something interesting: complex second also receives electrons, but these electrons comes from FADH2. And here it is the favorite exam question: that does complex 2 pumps proton? No. It transfers electrons only, no proton pumping. Because of this, FADH2 ultimately produces less ATP than NADH. Next, the complex third, that is called as cytochrome BC1 complex, or the cytochrome reductase complex. Here, third, it accepts the electrons from coenzyme Q, and again protons are pumped. Fourth complex is the cytochrome oxidase. This is the final station that electrons arrives here, oxygen accepts them, and water is formed. Without complex 4, respiration immediately stops. You will later see why cyanide poisoning is so dangerous. Finally, the fifth complex, that is the ATP synthase. Notice something: the complexes 1 to 4 mainly move electrons and create the proton gradient. Complex 5 does something completely different. It uses the stored energy, it manufactures the ATP. And think of the complex 1 to 4 as a workers filling in water tank, and complex 5 is a turbine generating electricity from the stored water. Students often write the all 5 complexes transport the electrons. That's incorrect. Only 1 to 4 transport the electrons, and 5th synthesizes the ATP. Let's see the next part. Here are the proteins required for the transport electron carriers for the ETC. The respiratory chain isn't just 5 complexes. Between these complexes, special carrier molecules that pick up electrons and deliver them to the next destination. Think of it little an Amazon delivery network. A package doesn't go directly from the factory. From the factory to your house, it passes through the several delivery centers. Similarly, electrons move through the series of the carriers until they finally reach to the oxygen. According to this, these are the important 5 carriers participate in the electron transport chain. First one is nicotinamide nucleotides. First carrier is NADH. Now it reaches to the complex first NADH dehydrogenase. And what happens here? NADH donates 2 electrons to complex first. After donating electrons, it becomes NAD plus, and just go back to the participate in metabolic reactions. But something else happens here. The energy is released while electrons move from complex 1 is used to pump 4 protons and from the mitochondrial matrix into intermembrane space. Why pump proteins? Because these protons will later drive ATP synthesis. These act as a charging battery. And second one is the flavoproteins. Now let's meet the second carrier. These are the flavoproteins which contains FMN and FAD. According to this, electrons from FADH2 enters through the complex second, and also called succinate dehydrogenase. Here is one important question: that does complex second pump protons? No. Complex second transfers the electrons to coenzyme Q, but does not pump H plus ions. That's the reason why FADH2 produces less ATP than NADH. This is one of the favorite university questions. Third one is coenzyme Q, that is ubiquinone. Now imagine you are driving on a highway and every road merges into one main highway. That exactly the coenzyme Q does. It receives electrons from complex first and second, then carries them to the complex third. Unlike the complexes, coenzyme Q is a mobile, and it can freely move within the inner mitochondrial membrane. Because it's lipid soluble, that's why it's often called the mobile electron carrier. Fourth one is the iron sulfur proteins. Now let's discuss the carrier that students usually ignore it. These are the iron sulfur proteins. They contain iron atoms that can easily switch between 2 oxidation states, and that allows them to transfer the electrons one at a time. In a complex first, FES is proteins receive electrons from the FMN and pass them into coenzyme Q ubiquinone. They are also present in complex third, so don't think they are separate complexes. They are electron transport centers within the complexes. Here, fifth one is cytochromes. Finally, we reach at this last group of the carriers, that are cytochromes. Cytochromes are proteins containing heme ions, and ion keeps changing the between Fe plus 2 and Fe plus 3 while transferring the electrons. According to this, cytochrome B passed to the electrons to C1, then C2, then A and E3. Finally, they reach to the oxygen. And now ask yourself which cytochrome is directly transferred electron to oxygen. That is the cytochrome A3, which is the part of the complex 4, and this is why the poisons like cyanide are also deadly. They block this final step. Now let's move to the next important concept, that is the oxidative phosphorylation. This sounds like a huge word, but it's actually very simple. Break the one word into two parts. Oxidation means the electrons are being removed from the NADH and FADH2. And phosphorylation means the ADP receives phosphate and becomes the ATP. So oxidative phosphorylation simply means the ATP synthesis that coupled with the electron transport. This process occurs at the complex fifth, also called as the ATP synthase. Located in inner mitochondrial membrane, without electron transport, ATP synthesis cannot occur. Without ATP synthase, stored proton energy cannot be converted into ATP. Both processes are tightly linked. Here now let's discuss the one concept students often memorize without understanding, that is P is to O ratio. What does it mean? P stands for phosphate used to make the ATP, and O stands for the atom of the oxygen is consumed. So P is to O ratio tells us how many ATP are produced per oxygen atom, reducing during the electron transport. Now look at the numbers. When electrons come from the NADH, they pass through the complex first, third and fourth. The total 10 protons are pumped into the intermembrane space to make the 1 ATP. Approximately 4 protons are required. Therefore, 10 divided by 4, 2.5 ATPs are generated. Now consider FADH2. Since it enters the complex second, skips the first, and since it enters into this second complex, only 6 protons are pumped, and here 6 divided by 4, 1.5 ATP is formed. According to this, ATP formation is coupled with three major energy conserving sites in the ETC, that between complex first and coenzyme Q, between the complex third, oxidation of cytochrome B by the cytochrome C1, and at complex fourth, cytochrome oxidase reaction. These are the sites where energy released from the electron transfer is conserved and ultimately used for the ATP synthesis. Only about 35% of energy is released during oxidation of NADH is trapped as the ATP, and the remaining energy is released as heat. And that's actually beneficial. Without this heat production, our body temperature could not be maintained. So even the lost energy has the important physiological role. Let's discuss the next part. Here, how does the pumping protons outside the mitochondria suddenly creates ATP? Can protons really produce energy? This is a brilliant question remained unanswered for decades, and until one scientist is proposed the revolutionary idea, that is chemiosmotic hypothesis. Once you understand this concept, ATP synthesis is become incredibly easy. Let's dive in. Before this theory, the scientists knew that the ETC chain produced the ATP, but no one knew how. Peter Michel proposed the simple yet but brilliant explanation. Let's see. Look carefully. This is the mitochondrial matrix. Let's see, here is the one mitochondria. It is the mitochondrial matrix, and it is the intermembrane space is here, and electrons move through 1, 3 and 4. Energy is released, and instead of wasting energy, the complexes use it to pump H plus ions from the matrix into the intermembrane space. What happens if more and more protons are pumped outside? Proton gradient develops, and one side now has a high concentration of protons and other side low concentration. Nature always tries to equalize the concentration, so these protons desperately want to come back into the matrix. But there is a problem, that the intermitochondrial membrane is almost impermeable to the H plus ions, and one way to back through the special channel, that is ATP synthase, and that's where the ATP is made. Here is the simple process. Rotatory model of the ATP synthase. Now comes the most fascinating molecular machine in the human body, that is ATP synthase. Scientists actually call it smallest rotary motor in the nature. Yes, inside every one of your cells, tiny motors are continuously spinning. But how does the proton movement rotate the protein? Think of a water mill. Flowing water rotates a wheel. Similarly, in the flowing water, protons rotate a F1 rotor, and that rotational movement is transmitted to the gamma subunit, and gamma subunit rotates inside the F1 head. Rotation changes the shape of the beta subunits, and these conformational changes convert ADP into ATP. And here, that's why the ATP synthase is called as rotary molecular motor. This is the diagram here. F1, F0 is here. This is the proton channel. Gamma shaft and beta subunits are there. One of the most favorite Viva question is that which subunit rotates? Gamma subunit is rotates. Which subunit synthesizes the ATP? The beta subunit synthesizes the ATP of the F1. Imagine ATP synthase suddenly stops, and even if oxygen is available, even if NAD is present, ATP cannot be synthesized. This slowly why the ATP synthase is absolutely essential for life. Now let's connect this topic to the medicine. If mitochondria fail, high energy organs suffer first, which is brain, eyes, heart, muscles. So mitochondrial diseases mainly affect these organs. These are the classic diseases like labor, hereditary, optic neuropathy. It is caused by mutations in the mitochondrial DNA. Patients gradually develop painless loss of central vision due to degeneration of the optic nerve. And other inherited mitochondrial disorders include mitochondrial encephalopathy, lactic acidosis, stroke-like episodes, and Pearson syndrome, mitochondrial DNA depletion syndrome, and Leigh syndrome. You don't need to just memorize every detail for first reading. Just remember defective oxidative phosphorylation mainly affects the organ with high ATP demand. Now let's imagine something dangerous. What happens if we block the electron transport chain? Electrons stop moving, ATP production stops, cells begin to die. Let's see where different poisons act. Complex first inhibitors between the NADH and coenzyme Q. Examples include that is rotenone, fish poison, amityl barbiturate, and pyridine A, that is antibiotic. This prevents the electron transport from complex first to coenzyme Q. Complex third inhibitor are there between B and C1, that is antimycin A antibiotic. This blocks the electron transport through complex 3. And complex 4 inhibitors now comes to the most dangerous group. These are the cytochrome oxidase group, carbon monoxide, cyanide, hydrogen sulfide, and azide. Because of the oxygen cannot receive the electrons, the entire ETC chain stopped instantly. Why does cyanide kill so quickly? Because it prevents the oxygen utilization ever when the oxygen is present in the body, in the blood, and the body literally suffocates at the cellular level. This condition is called as histotoxic hypoxia or the tissue asphyxia. This is one of the most important clinical correlation. According to this, the cyanide binds to the Fe+3 ions of the cytochrome A3 in the complex 4th, and as a result, electron transport stops and oxidative phosphorylation stops, ATP production ceases, and cells die rapidly. Organs with the highest oxygen demand, brain and heart, are affected first. This is called as the cyanide poisoning. Patients may present with the headache, weakness, giddiness, confusion, and difficulty breathing, and severe poisoning occurs coma, seizures, cardiac arrest, and death. And here's the treatment that according to this antidote is hydroxycobalamin, vitamin B12 derivative. It binds to cyanide to form cyanocobalamin, which is safely excreted from the body. Which is the complex is inhibited by the cyanide? The answer is the complex 4th, cytochrome oxidase. Imagine this: the electron transport chain is working perfectly. The electrons are flowing, oxygen is available, protons are being used, pumped, and ATP is still not being produced. Sounds impossible. Actually, this happens in the presence of uncouplers. Today, we will understand why this happens and where it is useful in our body, and finally master the malate aspartate shuttle. Let's begin. First, let's understand what is uncoupling. Normally, two processes are tightly attached, that is electron transport, and another one is the ATP synthesis. Electron transport creates the proton gradient, and ATP synthase uses the gradient to make ATP. These two processes are coupled. Now imagine someone cuts the connection between them. Electron transport continues, oxygen is still consumed, but ATP synthesis stops. This is called as the uncoupling. Imagine filling a water tank. Normally, water flows through turbine and produces the electricity. Now, someone makes a big hole of the wall of the tank and water leaks out of the turbine. Electricity is never generated. Exactly the same thing happens inside the mitochondria. Instead of passing ATP synthase, and protons leak directly back into the matrix, and the proton gradient disappears and no ATP is produced. The stored energy is released as the heat. Remember, the electron transport continues, oxygen consumption continues, the only ATP production decreases. This is the most favorite university exam questions due to uncouplers. Here are the examples for the uncouplers. This is first one is the DNP, that is 2,4-dinitrophenol. This is the most famous uncoupler. It is the small lipid soluble molecule and it easily crosses the inner mitochondrial membrane carrying the protons. Instead of following protons, it passes through the ATP synthase and DNA carries them directly back into the matrix. And results no ATP, excessive heat production. Years ago, people used the DNP as a weight loss drug. Why? Because nutrients continues to be oxidized but the energy was lost as a heat instead of being stored as ATP. People lost weight rapidly. Unfortunately, many developed severe hyperthermia and some even died also. That's why why DNP is no longer used. Let's discuss the another dinitocresol. Here, pentachlorophenol, trifluorocarbonyl cyanide, and phenylhydrazone and hydroaspirin. You don't need to memorize all of them, just remember the DNP as a prototype. Now here's the something interesting. Now, not all uncoupling is harmful. Our body intentionally uses uncoupling to generate heat, and best example is thermogenin. Here is the best example, that is the thermogenin. Here, it is important in the inner mitochondrial membrane of the brown adipose tissues. Brown fat abundant in the newborn babies and hibernating animals. Why is the brown fat is special? Brown fat contains numerous mitochondria and these mitochondria have thermogenin. Thermogenin follows the proton to return directly into the matrix without passing ATP synthase. Therefore, ATP is not produced. Instead, the large amounts of heat is generated. This process is called as non-shivering thermogenesis. Newborn babies cannot shiver efficiently, so they maintain body temperature using brown fat. This is the favorite Viva question. Let's discuss about the ionophores and other inhibitors. Let's quickly discuss the few additional points. Ionophores are lipid soluble and they transport ions from across the biological membranes, include the valinomycin and gramicidin A and nigericin. They mainly transport potassium and disturb the ion gradients, and there is another oligomycin and atracetyloside. Let's discuss about the last topic from the notes. Here's the question that during glycolysis, NADH is produced in cytoplasm, but the electron transport chain is inside the mitochondria. Can NADH directly cross the inner mitochondrial membrane? No. The membrane is impermeable to NADH, so how the electron transported? Through the malate aspartate shuttle. Here in the step 1, in the cytosol, oxaloacetate accepts the electrons from cytosolic NADH. OAA becomes the malate, and at the same time NADH becomes NAD, and now becomes the malate can cross the inner mitochondrial membrane. Here is the step 2 in the mitochondria. Malate is converted back into oxaloacetate by the mitochondrial malate dehydrogenase. During this reaction, new mitochondrial NADH is produced, and this NADH enters the electron transport chain. Since it enters the mitochondrial NADH, it generates the 2.5 ATP. And step 3 is the aspartate formation. Here, but oxaloacetate cannot cross the membrane. So what happens? The oxaloacetate undergoes transamination with glutamate. It becomes the aspartate, and aspartate moves back into the cytosol. Here, and another transamination reaction regenerates the oxaloacetate. Here, according to this, the liver and heart mainly use the malate aspartate shuttle. Each cytosolic NADH ultimately produces 2.5 ATP. And students often confuse this. Remember, the malate aspartate shuttle 2.5 ATP, another shuttle glycerol phosphate shuttle 1.5 ATPs. And here we have covered the whole ETC. These are the concepts that you will use repeatedly in biochemistry, physiology, pharmacology, pathology, and medicine. If this lecture helped you truly understand the concept instead of just memorizing it, subscribe because we are covering other chapters also. Thank you.